The following program is brought to you by Caltech.
Good evening, and welcome to tonight's Watson Lecture. I'm David Terrell from the Division of Chemistry and Chemical Engineering at Caltech, and I have the high privilege and distinct honor of introducing tonight's Watson lecturer, Jackie Barton. I was asked, though, before introducing Jackie, to remind you that the Watson Lecture Series is an ongoing series, and to alert you to the next Watson Lecture, which will be held on February 17th. It will be given by Aris Razakis, who is Division Chair in Engineering and Applied Science, and Aris will talk to us about intersonic earthquakes, what laboratory earthquakes teach us about real ones. Tonight's speaker is Jacqueline Barton, who is the Arthur and Marion Hanisch Memorial Professor and Chair of the Division of Chemistry and Chemical Engineering. Jackie is a native New Yorker. She was educated at Barnard College and at Columbia University. She did postdoctoral work at Bell Laboratories and at Yale before taking her first independent position at Hunter College in New York. She was called back to Columbia a short time later and stayed there until 1989, when she was persuaded to join the Caltech faculty. Over the course of her career, and in particular during her now 20 years at Caltech, Jackie has made a remarkable series of contributions to our understanding of the chemistry of the nucleic acids and shown us that the chemistry of DNA is much richer than any of us had imagined it to be. Her work has been recognized in many ways, most notably perhaps by her election to the National Academy of Sciences and to the American Philosophical Society, and by the awarding of numerous honorary degrees, including one from Yale University, Williams College, and Kenyon College. Her lecture tonight is on the subject of DNA-mediated signaling. Jackie?
Thanks very much, Dave, and thanks all of you for coming after the State of the Union. Sort of feel it may be a step down, but I'll tell you a little bit about the state of some chemistry. What I want to talk to you about tonight is DNA, the stuff of our genes. But what I really want to talk to you about is the chemistry of DNA. And in particular, we've been learning about one of the unique characteristics of DNA chemistry, and how more recently we've been focused on what the biological implications of that chemistry might actually be. So I'm going to start out in first gear and get everybody thinking about the structure of DNA and the properties of DNA, and then we'll move it up to second and third gear, and right up to telling you something about some of the recent experiments that we've been doing that we're pretty excited about. So let me begin with a few pictures of DNA. And what's shown here are fluorescent micrographs of the 23 pairs of chromosomes of DNA. DNA encodes everything that makes us what we are, that makes you different from you, from a plant, from a bacterium. And when we think about DNA, this library of the cell, I think about pictures that look more like this. This is the structures, the chemical structures of DNA. DNA is a polymer, just like polyethylene is a polymer. This polymer has repeating units that each contain a sugar, a phosphate, and a nucleic acid base. And these phosphates are negatively charged, so it's a negatively charged polymer, and it goes sugar, phosphate, sugar, phosphate, sugar, phosphate. Now, as I said, we have these 4 nucleic acid bases, adenine, guanine, cytosine, and thymine. And so really, that represents the alphabet of our DNA, of our genome, and it's an alphabet with 4 letters. Now, when you take one strand of DNA and you associate it with the other strand of DNA, you get DNA base pairs. And it turns out that A pairs with T and G pairs with C. And when those 2 strands come together, they form this beautiful double helix. Let me show you another prettier picture of the double helix. This encodes 30 base pairs of DNA, 3 turns of a helix. And I've got the, let's call it the Watson strand in pink. Maybe it's the Crick strand is in blue. And as you see, we've got 4 different colors, and green always goes with red, and yellow always goes with blue, A with T, and G with C. Now this distance, this represents, as I said, 30 base pairs, and that would be a millionth of a centimeter long. It's actually pretty short. But in each one of our cells, in every one of our cells, we have 3 billion base pairs of DNA. So if you strung it all out, it would represent about a meter's worth of DNA in each one of our cells. Very skinny, but about a meter's worth. And that does indeed represent the library of our cell. And in some cells, some of the information's being read. In your eye cell, what it takes to be an eye needs to be read. In your skin cells, what it takes to be on skin is being read. And that information is being read and converted into RNA, converted into protein, the machines that carry out the reactions within your cell. So the analogy is usually done, often done, that we think of DNA as the library of the cell. And when you express a gene, you go and find a book in that library. You have to find the promoter region, the region of the DNA that tells you what's in the book. And then you'll open up the book, take out a page, maybe Xerox that page. That's like making the RNA. Take that page down to the machine, the factory that converts that information into a protein. We call it the ribosome. But that's all I want to talk to you about right now, about DNA as the library. Because you'll notice in the picture I also have a pencil. And I really want to talk to you about the pencil. In fact, I want to talk to you about the graphite point in that pencil. So let's talk about why that is. So here is a schematic view of graphite. So graphite is made of all carbon atoms. And when you have graphite, it's arranged in planes, in sheets. And I made them yellow and red so that you could distinguish one plane from the next. And when you write with your graphite pencil, a little bit of that sheet is being sloughed off, and that's how you get the pencil mark on the page. In any case, in each of these planes of carbon atoms, you have the carbon nuclei, and you have electrons that are delocalized, that aren't just associated with one carbon nucleus, but they're moving around that sheet. And it turns out, if you were to apply potential across one of these sheets, the electrons would move in the direction of the applied potential, and that's conductive. So graphite is a conductive material. Now, it also turns out, if you've got one sheet with these planes of delocalized electrons, they're interacting with the next sheet, with the next plane of delocalized electrons by weak interactions. And it also turns out, if you apply a potential along that direction, because you have these delocalized electrons, you'll even get conductivity along that direction. Not as much. The electrons won't migrate nearly as easily as they would in the perpendicular direction, but you will in fact get some conductivity, particularly if you pull some electrons out so that you've got holes, you've got room for the electrons to travel. Okay. Now, it turns out that's a general characteristic of solid-state materials that have these delocalized pi electrons. That's what they're called. Now, some of you are probably looking at your watch already and saying, I thought we were going to talk about DNA tonight. Okay, so let me tell you what this has to do with DNA. So here's another picture of DNA. But in this picture, all the base pairs are the same color because all of the base pairs have these delocalized pi electrons. And instead of representing the sugar phosphate backbone, I've schematized that as a ribbon to focus your attention on this stack of pi stacked base pairs, sort of like a stack of copper pennies. And if you look down the helix axis, if you look down the perpendicular direction, they really are all stacked right on top of another. And a large measure of the stability of double helical DNA is derived from that stacking interaction, from how they stack together. And so the question arose, essentially just 3 years after Watson and Crick first proposed the structure of DNA. It was suggested by Albert St. Georgi. It was suggested that maybe like graphite, like these solid-state pi-stacked materials, DNA might also have some conductivity properties along that long direction. Now, as I said, it was first suggested by Albert St. Georgi, but I think it was probably best articulated by Ellie and Spivey, who were two biophysicists working in England in the early 60s. And they said the bases are thus arranged like a pile of coins along the helix axis, and their interplanar spacing of 3.4 angstroms is similar to that for graphite. It seemed, therefore, reasonable to suppose that a DNA molecule might behave as a one-dimensional aromatic crystal and show pi conductivity down the axis. And so that was the question. So now, maybe some of you in the audience can think back to the 1960s. I certainly can. But now in the 1960s, it was hard to do that experiment. How might you see whether or not DNA could conduct? Well, what they did was they'd crack open cells. The physicists would go to their neighbors, the biologists. They would harvest some cells. They'd crack them open. They'd isolate the DNA. They centrifuged it down into a gelatinous pellet. And they took 2 alligator clips and put them on either side of the pellet. Yeah, so some of you are laughing. Yeah, so it turns out, not a great experiment. DNA isn't oriented. Remember, it's negatively charged, so it has all these positive counterions associated with it. It's not quite clear what you've got, where the alligator clips are going. And so it seemed to us, now actually almost 20 years ago, that this was a question for chemists. Because chemists could go into the lab, and what we did was we made a molecular alligator clip experiment. We could go into the lab and synthesize a piece of DNA and attach 2 metal complexes, one at each end, that also had pi electrons, and so they were pi stacked right in with the DNA, and ask whether or not electrons could flow from one metal center to the other metal center. And in fact, the experiment was a very straightforward one because it turns out that the red metal complex, which is a ruthenium complex, could glow, it luminesced. But if you had the rhodium complex bound on the other side, that glow was quenched. It didn't glow anymore. And it didn't glow because that excited state was short-circuited because the electron moved from the ruthenium to the rhodium. And so that, it turns out, was a pretty provocative experiment because it suggested that, in fact, you could have an electron transferred from one center to the other center, and in fact, over quite long molecular distances compared to what people had seen before. So that really started a flurry of activity in our lab and labs pretty much around the world, making various assemblies to look at long-range electron transfer, to look at how electrons could move through the DNA helix. And over the past 20 years, we've learned some lessons. And I'll sort of summarize the most important ones. And the first is that electron migration through DNA occurs over long molecular distances. It can go a long way. And there's a shallow distance dependence. It doesn't matter how long it is, it can keep on going. As long as the bases are well stacked. sort of like a stack of copper pennies. And if one of the copper pennies were tilted in some way, that would interrupt the conductivity of the stack. And similarly, I think in fact quite similarly, transport through DNA is interrupted by perturbations in base stacking. And that's what we learned. Okay. So let me give you some examples of that and the kinds of experiments that we did. Well, one thing was we began to think we don't really need two metals at either end, or two complexes at either end, a donor and acceptor that would inject an electron and pull one out. What about a reaction directly with DNA? Could we take an electron right out of a DNA base? It's easy to oxidize DNA bases, to take electrons out of them. Could we use something, a photooxidant, which could really extract that electron? The other thing that people in the field tend to do is, instead of talking all the time about abstracting electrons, they also talk about injecting holes, which is the same thing, sort of the opposite, but same thing. Anyway, so we did this experiment. We tried to extract an electron from DNA, and the base in DNA that's easiest to pull an electron out of is a guanine, and particular guanine doublets. If you had two guanines stacked up next to each other, the left hand, the 5' guanine, is easier to pull an electron out of. And it turns out we could do this experiment a lot of different ways with a lot of different sequences. And Megan Nunez, who was my student at the time, she's now a professor at Mount Holyoke, she still actually holds the world's record for demonstrating long-range damage to DNA, long-range oxidation of DNA, because she made an assembly and attached a metal complex at the end of it, and she was able to show damage to a guanine 200 angstroms, 20 nanometers away from the metal center. Now, I'm not going to convert that to centimeters for you because you're not going to be impressed. But on a molecular scale, it's like throwing a Hail Mary pass. It's really far away in terms of that molecular way of thinking about it. Okay, so what about this dependence on base stacking? Well, we did another experiment where we said, okay, if it's sensitive to stacking, what if instead of having an A opposite a T, we synthesized a piece of DNA so that we had a C opposite the A, sorry. Instead of a T opposite the A, yeah, we had a C opposite an A. That would be a mismatch. And it turns out, in this case, we used an organic fluorescent molecule. It's quenched because of electron transfer, because it's short-circuited. But if instead we have a mismatch, it's not quenched anymore. No short-circuiting. It's sensitive to that mismatch. And just to calibrate you, a mismatch isn't a really big change in the DNA. Here are structures that we know of, static structures obtained from shooting X-rays of crystals of different sequences of DNA. In this case, it's all Watson-Crick base paired. A's are all with T's and C's are all with G's. In this case, we have a GA mismatch. And a GA mismatch, you've got those two double rings. They're actually stacked pretty well. Here's the CA mismatch. Now, you wouldn't have noticed if I didn't color code them white. It's a very subtle perturbation. Turns out they move around a little bit more than if they're well base paired. But the stacking is only a little bit off. But just that little bit is enough to turn off this electron transfer reaction. It's really quite remarkable. Now, as we've been doing these experiments, invariably, there would be a physicist in the crowd who would raise their hand at the end of one of my seminars and say, but what is the conductivity of DNA? And it turns out that's a hard experiment to do. A lot of physicists have tried to do that experiment, but they, in order to use sophisticated techniques to try and get that molecular conductivity, that conductivity of a single molecule, and measure what it was, they had to submit the DNA to tortuous things. 0 degree Kelvin, really cold. Usually no water. Now, I just told you, it's really sensitive to stacking. So those experiments were almost as funny as the alligator clips on the pellets, okay? But then I was reading a paper recently in Science Magazine by Colin Nuckolls, who's professor at Columbia University. And Colin had devised this wonderful way to measure molecular conductivities of single molecules by creating devices that could allow him to measure conductivity. And in this device, he would start with a carbon nanotube. Now, a carbon nanotube is similar. It's not the same, but it's sort of similar to graphite. It's only made of carbon, and it's sort of like sheets of graphite, sort of the long way. And so it is quite conductive. And then what he would do was he'd use an oxygen plasma to burn a little gap, a very small gap, that's the gap size, it's very, very small, into this carbon nanotube. And as you burn carbon, you're going to generate what are called carboxylic acids, will generate functional groups inside this carbon nanotube to which we can attach something else by covalent bonds. So I know exactly what the chemistry is, and I know exactly what I have. And when you do that, you can then measure the conductivity in that device relative to what it was before you burnt out the little gap. And so I called up Colin on the phone, and we did the experiment. We could attach one strand to one end of the gap and another strand to the other end of the gap, or we could just attach a single strand and float in the complementary strand. And in both cases, what we saw was before we had a gap, when we had just the conductivity of the carbon nanotube, we had a certain amount of current flow. After we burnt out the gap, we had no flow. We broke the wire. But then if we connected it with the DNA, we could see that some current returned. And that was true both with the single strand attached or with the duplex attached on either end. But having just one single strand attached lets you do what I thought was a pretty neat experiment. We could start out with well-matched DNA, and then in this device, and I should say the reason why I called Colin in the first place was you could do these experiments in water. It was a robust system. And so we could then flush out that complementary strand because it was only attached by non-covalent interactions, by a little bit of hydrogen bonding and stacking. You could just dilute it with water and get that strand away, and then float in another strand that maybe had a mismatch in it. So this is a CA at this spot, and it was a TA before. You could float that out and put in another mismatch, maybe a GT mismatch. And so it turns out if you measure the current for these different devices, when it's well matched, it had a high current. When you had a CA mismatch, the current went down, you interrupted that current flow. GT mismatch, same thing, and then if you floated back in a well-matched strand, it would come back up. So we could do this reversibly. We knew then that it really corresponded, the mismatch interrupted current flow, but if you had a well-stacked DNA, it worked just fine. And then we could carry out measurements, quantitatively determine versus the carbon nanotube what it looked like, what the conductivity might be. We could estimate if we had a piece of graphite in that gap, what the current would be, what the resistance of that material would be. And it turns out from experiments that physicists had done before, they couldn't agree. They were varied by a factor of 100 billion, sort of like the economy. But if you looked at graphite, so graphite has a very low resistance, it's highly conductive in the parallel direction. If you had graphite in the perpendicular direction, the analog of DNA, that had a resistance of 3 × 10⁵ ohms, just like well-matched DNA, essentially just like well-matched DNA. And if you had a mismatch, then it was a 300-fold higher resistance, or 300-fold decreased current flow. Okay? So that was the result. We now really knew that the analogy to graphite was a good one. Okay, so now you can take DNA and you can hook it up to things and see what happens. And so we would take DNA, double-stranded DNA, and have a little linker and attach it to a gold electrode and see, can I get current flow? You can measure the current flow, and we would measure the current flow to some probe that we had attached to the top. Now, these are funny-looking diagrams, but don't stay with me on this. These are just current as a function of potential, so we would increase the potential on the gold surface, and when we had enough driving force, enough oomph, then the electrons would flow from the gold surface to the top of the DNA. So as we varied the potential, nothing happened. Then when we had enough driving force, enough potential, we'd see current, see a blip, and then we'd cycle it back. We'd put the potential the other way around, and so then the electrons would flow from the reduced probe back to the gold surface, another blip, and you come back, okay? But what if I now change the T to a C, keeping everything else the same? No current flow, no blip. It was remarkably sensitive to the presence of an intervening mismatch. Now, why do I care so much about mismatches? Well, it turns out that mismatches can be predictors of disease. Because now we know from the Human Genome Project, we know essentially for lots of different proteins what mutations cause problems, what single base changes in those proteins, in the genes that encode the proteins, lead to a predisposition to colon cancer, let's say. Or sickle cell anemia is probably the classic example of a single base mutation causing a change in the encoded protein which causes disease. So if I now have the wild type sequence, the good sequence, the sequence that encodes the protein that works, and let's say at a particular position in that sequence of DNA, I have a GC base pair, and I want to test your DNA and ask, is your DNA encoding the right sequence? Do you have a possibility of sickle cell anemia, or does your child have the possibility of having sickle cell anemia? And so you test that piece of DNA. You can test it by taking that sequence. If you have a single base mutation, let's say instead of a G you have an A, then if I take that piece of double-stranded DNA, mix it with the test DNA, heat it up so that the strands separate, and then they all come back randomly together, I will be generating 50% mismatches. So mismatches, if I can detect a mismatch, I can detect a single base mutation. But think about it, in a long piece of DNA, it's non-trivial to detect a mismatch, but this chemistry allows you to do that. And in fact, we could detect all single base mismatches, even the GA mismatch that was so nicely stacked in that first picture, all single base mismatches, no matter what was around them, electrochemically, with a little electrical device. And that allowed us to make little DNA chips where you could do mutational analysis. And that ends up being a very sensitive sensor and a very cheap and easy sensor to detect mutations in DNA. What's absolutely remarkable about this chemistry, and it was surprising every time we would do an experiment on a new sample and look at it, how sensitive it is to little mistakes in the DNA. So here are structures, chemical structures, of different lesions, different mistakes, different damage that can occur to your bases in DNA. You can just have an extra oxygen on adenine. That's called 8-oxoadenine. And if you put it in our system and you look to see whether or not you get current flow or charge accumulation, with OXO-A opposite a T in a long sequence of DNA, there's no current flow. If you had a regular AT, you get a lot of current flow, a lot of charge accumulation. So these very small changes are enough. Just a single atom difference is enough to turn off this chemistry. It also turns out some proteins, when they bind to DNA, cause a change in stacking. Some don't do much, some make a big difference. And so we could take DNA, put it on these gold surfaces, and then add proteins and ask whether or not that turned off the current flow. Here's an example. This is TATA-binding protein. It's a protein that's involved in turning genes on. Essentially, it helps to sort of bend the DNA, kink the DNA at the spot where then all the apparatus, polymerase apparatus, comes along and reads that genetic information. And when it kinks the DNA, you add the protein to the DNA, it turns off the current flow, versus when you had the DNA without having the protein bound to it. So it is a very sensitive assay. And just to give you a sense of things, these are recent experiments that we're doing. We're now making multiplex chips. Jason Slinger, Natalie Murin, Ilan Gorodetsky are fabricating these chips, putting different probes on different spots on the chip, looking at protein reactions on DNA, looking at very long sequences of DNA. Jason's actually going for the world's record, and I think he's got it. So there are all sorts of interesting sensors and real DNA chips that you can make using this chemistry. But what we've become really interested in is asking the question, if this is such interesting and good chemistry, nature tends to take advantage of what it's got. And this chemistry allows you to carry out a reaction over a very long distance. And when proteins bind to DNA, they can modulate that reaction. So maybe nature uses it. Maybe this goes on within the cell. So we actually started thinking about this several years ago, a while ago. Before I tell you about that, when you think about where nature might use this, I got to talk to you about stress. I could really tell you about stress, because what Dave didn't tell you was he used to be chairman of our division, but now I am. So I know, he's smiling and I know about stress. But I'm not talking to you about that kind of stress. I'm talking to you about oxidative stress. In fact, it might be related in some respects. And oxidative stress is associated with aging. It's associated with Alzheimer's. It's associated with many diseases, with cancer. And when we talk about oxidative stress in terms of what goes on within the cell, it's quite complicated. It's a complex operation, but it turns out in each one of your cells, you take oxygen and you burn fuel and you generate energy. Now, when you do those reactions, and there are many complicated reactions that allows that to happen, this shows some of the various pathways. You start with oxygen, and you're actually supposed to end up with water. And electrons go and get used. About 4 electrons are shuttled to the oxygen in the course of the reaction to store the chemical energy and burn the fuels. But if you are undergoing oxidative stress, if things aren't working quite right, then in fact, some of these electrons get a little leaky, and you start generating things like peroxide instead of going all the way to water. And it turns out the electrons that get shuttled to oxygen in the course of these reactions, the shuttlers who are carrying out this chemistry are usually metals, coppers, irons in particular. And it turns out that when the cell is undergoing oxidative stress and when you have aging, then that gets a little leaky as well. And when you take peroxide and you take iron or various metal ions, those electrons will get together and generate something called a free radical. And a free radical is just the way it sounds. It really wreaks havoc. That free radical wants an electron so bad, it's going to abstract it from things. And often, it'll abstract it from membranes. And sometimes, often, it will abstract it from your DNA, because remember, your DNA bases, it's easy to pull electrons out of them. And so we started to ask the question, if you abstract electrons from DNA at point A, we know that you can get migration of electrons through the DNA. Maybe electrons get shuttled to some spot. Maybe there's a spot where all of that damaging oxidative damage, essentially through holes, electron holes, gets shuttled to be sort of the junk area, the area that's the damaged spot within the cell. Maybe some parts of the genome are protected from damage, insulated from damage, so the electrons and holes never go there. And so we started to ask those kinds of questions. The first simple kind of question we asked is, if we took one of our metal complexes that abstracts electrons from DNA and incubate them with cells or with cell nuclei, what kind of damage do we see? Well, we ended up seeing damage on Gs, the 5' G of Gs, consistent with the idea that electrons were being abstracted. But then it turns out in your cells and in my cells, the DNA isn't just naked DNA. The DNA is wrapped in nucleosomes, those fluorescent micrographs that I showed you of chromosomes in the first slide. That represents all DNA wrapped around proteins in a magnificent way that we don't totally understand, that packages that DNA, much like books on a shelf in a library, although I think probably far more complex than that. Anyway, there are some crystal structures of DNA wrapped around these histone proteins, like that which occurs in a chromosome. And so we ask, if the DNA is wrapped into these nucleosome structures, are they protected from damage? Maybe they're not going to get damaged as badly. Well, it turns out that they do. So we took a metal, we stuck it at the end of one of these pieces of DNA and wrapped it around the histones, and it turns out that the pattern of damage was the same as if we didn't have them wrapped around the histones in the first place. So when we think of DNA being packaged and protected from damage within the nucleosome, they're not protected from this kind of damage, from long-range damage through electron transport. And then we started asking another question, and that is, when we think about this electron moving through DNA, we're thinking of DNA sort of like a wire. Maybe you could use this chemistry for long-range signaling. Maybe, in fact, and the chemical way to ask that question is maybe you could use this chemistry to oxidize or reduce a DNA-bound protein. Maybe a protein bound to DNA could have an electron go into it through the DNA, or an electron be pulled out of the protein from the DNA, and then that changes the chemistry of the protein. It's a way to signal a reaction at long molecular distances across the genome to a protein, and maybe turn on the reaction of the protein. So here's an example of one such protein that we've looked at recently. This is a structure of the protein SOXR that's bound to DNA, and SOXR is in bacteria, and it turns out that SOXR is the protein that's the first sensor of oxidative stress within the bacteria. And so when the cell is undergoing oxidative stress, SOXR ends up getting oxidized, an electron comes out of it. One doesn't know how, one doesn't know where, but it comes out of it, and then that oxidation of SOXR turns on essentially the whole library of genes, brings the books out of the shelf that encode the genes for responding to oxidative stress within the cell. Where does that electron get abstracted from? It's not known, but it is known that SOXR, there aren't a lot of SOXRs within each bacterium, and the SOXRs are bound to DNA. They're always bound to DNA, oxidized or not. But if they're oxidized, they have an electron abstracted, that turns on the response. So we thought, well, this is a perfect example. This is a perfect thing to think about. Maybe we could use a metal complex, shine light on it, and instead of just abstracting the electron from the DNA, maybe through that migration then, that DNA with a little hole on it because it's lost its electron, then the SOXR could fill that electron hole, abstract the electron, and turn on SOXR. And so Paul Lee went in the lab, and he showed in a test tube that you could do that. Got rid of all the oxygen, had a glove box without any oxygen, had a little fiber optic cable so he could shine light on it, and he showed that he could turn on transcription, that you could use the whole polymerase machinery. In addition to this, if the SOXR was oxidized through the DNA, then you could turn on the reaction. You could signal to SOXR through the DNA. Now, it turns out in us, in eukaryotes, not in bacteria, we don't have SOXR. We have other proteins and protein pathways, and it's a lot more complicated than it is within the bacterium. But one of these proteins is p53. And it turns out that p53 is referred to as the guardian of the genome. And that is because p53 is a DNA regulatory protein, but it binds to all sorts of different spots in the genome. It essentially reads all sorts of books on the bookshelf. And depending upon the cellular stresses, p53 may essentially be binding to one book, reading one section to respond to one kind of stress, or reading another book to respond to another kind of stress, binding to another region of the DNA. And how do we know, how does p53 know, whether to bind to one sequence of the DNA or another sequence of the DNA? It wasn't really clear. And it turns out that p53 binds to some sequences, turns on some proteins, some gene expression of some proteins, under conditions of oxidative stress. And so Katie Augustin in the group did an experiment first where she asked, well, maybe p53, like SOXR, gets turned on because under oxidative stress, it gets oxidized in a DNA-mediated reaction, that there's a signal from the guanine radicals that are generated to p53 to turn on p53. So we showed it in a test tube. We took one of our probes and attached it to the end, shine light on it, abstracted an electron from p53, and we showed that you could do that in a test tube. We also showed that you could do that in a cell by using an oxidant bound to DNA, and then we also showed that it only got oxidized when it was bound to some promoter sequences and not to other sequences, consistent with the idea that this was a way to signal across the genome that the cell was undergoing oxidative stress and respond to it through chemistry, through a different reaction, through oxidation, through DNA. Okay, last story. Remember, we have this reaction that is extraordinarily sensitive to mismatches in DNA and to lesions in DNA, single atom changes in DNA that occur when DNA bases get oxidized. In fact, you got to eat your broccoli, because if you don't eat your broccoli, you get more of this kind of damage. That's what antioxidants are about. Antioxidants are things that get oxidized instead of your DNA when you have this free radical damage. Okay, so now you have the damage, didn't eat the broccoli. You've got the damage, which is in fact occurring in each one of our cells, broccoli or not, all the time, every day. Each cell undergoes, I think, on the order of 1,000 incidents of this kind of damage. And that damage then has to get repaired. And it turns out we have an exquisite machinery of proteins within each of our cells that are searching for this damage, finding it and fixing it. Now, it turns out we understand today a lot about the chemistry of how these proteins fix the damage when they find it. But we didn't really understand how they might find it. And it is, hence the painting by Pizarro, sort of like looking for a needle in a haystack. Especially when you think just one of those oxygens and one of those bases is enough to cause a change. How do the proteins find it? Well, we thought it might use DNA charge transport chemistry. This chemistry we've been talking about. And it also turns out, interestingly enough, that a subset of these repair proteins have little iron clusters in them. Here's one of these little iron clusters. And it turns out all of these base excision repair proteins have what are called iron-sulfur clusters in them. And iron-sulfur clusters are known in other protein families to carry out electron transfer reactions, but nobody thought they'd be doing that within DNA. And so we thought we'd look at that. And so we took our DNA-modified electrodes, and we added these proteins that had the iron-sulfur clusters in them, and looked to see whether or not we had a blip, whether or not we could put electrons in or take electrons out, and we could. And it turns out we found that for all the proteins bound to DNA, it took the same driving force to take electrons out or put electrons in. And it also turns out that from what we saw in terms of our measurements, that when the protein was bound to DNA, it was easier to oxidize, take an electron out, than when it was free in solution, when it wasn't bound to DNA. Okay, so how do we think this works? Here's the idea. You've got the protein in solution in the cell, in the cytoplasm, whatever. And the protein's in the cell. In this case, these proteins, there's an analog in bacteria, there's an analog in you and in me. Some proteins, it turns out, go after one kind of damage. Other proteins go after another kind of damage. All of the proteins are at very low concentrations within the cell. A serious needle in the haystack problem. Okay, so let's say one of these proteins has a certain amount of electrons associated with it, and it binds to DNA, so it's easier to get oxidized. So it's ready to get rid of its electron, and it takes that electron and it sends it to another one of these proteins that also have an iron-sulfur cluster in them through the DNA, and then that protein, because it's now been reduced, it has an extra electron, it doesn't bind as tightly to the DNA as when it was oxidized. That came out of our measurements. And so that protein falls off the DNA and it goes someplace else. Okay? Now what I've just described for you is a scan of this region of the genome. The DNA was fine. There were no mistakes. So you could do the electron transfer. It had good conductivity. And so the protein fell off and it went someplace else. It's sort of like two telephone repairmen looking for a mistake in a wire. If they can talk to each other, everything's fine. If they can't, you got a break in the line. And we think that's the way it works. Now, if the protein then binds to a region of the DNA where you have a mistake, then it can't do the electron transfer reaction, and so slowly it's going to diffuse, bump into the spot, and find it. You've converted the needle in a haystack problem to a much smaller problem. You've redistributed the proteins in the vicinity of the lesion. So how do you test this? Well, we used a lot of experiments, different kinds of experiments to test this, but one experiment that you might understand is the idea that if I now have strands of DNA that don't have a mistake and other strands of DNA that have a mismatch, not even the thing that this repair protein fixes, but just a mismatch, we ought to see that the proteins, like the telephone repairmen, are going to redistribute themselves on the strands that have the mismatch. Does that make sense? So that's a pretty straightforward experiment. And so Pam Zons went in the lab, and she made pieces of DNA. This is the not straightforward part. She made big pieces of DNA with one single base mismatch in it. And these were long pieces of DNA, too big to synthesize on a machine. But she could do that, and she made big strands and bigger still strands. And the short strands didn't have the mismatch, and the longer strands did have the mismatch. And then she could use atomic force microscopy that allows you to make pictures, to measure DNA And so here is an atomic force microscopy view of DNA. And on this DNA is a little protein. Here's sort of a 3D view of it. Here's a little piece of DNA. There's the protein on it. And she took these samples, and she literally counted how many proteins were on the mismatched strand, the long strand, how many proteins were on the matched strand. Straightforward experiment. And it turns out there were more proteins on the mismatched strand. They really did redistribute, like the telephone repairman, in the vicinity of the lesion onto the strands with the mismatch. And then she added peroxide, oxidative stress. And the more peroxide she added, the more redistribution onto the mismatched strand was seen. And that's what you want. Under oxidative stress, you need repair. You want repair to be more efficient, and that's what we saw. And then the wonderful thing about Caltech students is they're great at saying, can we calculate this? And Joey Generu came into me one day and he said, can we calculate how long distance you have to get an electron scanning the genome in order for this to be of any utility? Remember, you've got billions of base pairs. Do you have to have megabase distances for electron transfers through DNA? We've only demonstrated it over a couple of 100 base pairs. So he calculated first that if you don't have electron transfer and you just essentially looked at each base along the strand, sort of like taking out all the books in the library and going through the pages to see if there was a tear in one of them, it would take a long time. It would take longer, in the case of E. coli, than the lifetime of the E. coli. So that's unacceptable. But then if he just incorporated electron transfer scans, even over a couple of 100 base pairs, you improve the efficiency very rapidly. So you're now scanning over regions. You're not just scanning base by base by base. And the interrogation is very quick. And so it makes sense. This is not an outrageous thing to be suggesting. And then finally we said, well, this is all well and good, and chemists like experiments that you can do in test tubes, but does this happen inside a cell? Can we provide any evidence that an electron will travel from one protein to the other protein through the DNA, signal each other inside a cell? Now that's a non-trivial experiment. But what we did was we collaborated with my colleague Diane Newman and carried out a genetics experiment. Because if we have these two different proteins, some of which are repairing one lesion, some of which are repairing another lesion, you can have a genetic assay for the efficiency of repair of one lesion versus the efficiency of repair of another lesion. And if we consider our model, let's say we have one of these proteins, it's called Endo III. When it binds to DNA and it sends electrons to another one of these proteins, that's called Muty dissociates and goes and finds the lesion. So Endo III is helping Muty find its lesion. So I can genetically knock out Endo III, and if I can assay for how well Muty fixes its lesions, if I've knocked out Endo III, I should get more mutations that Muty isn't fixing. Muty isn't going to work as well. Okay? And that's what we did. And it turns out that happens. So if you have the happily functioning E. coli, you only have a very low number of mutations with Muty, the kinds of lesions it generates. If you knock out Endo III, the helper protein, the other repair protein, the number of mutations go up. If we add back in DNA that encodes a wild-type Endo III, the mutations go back down again. And if, in fact, we encode a plasmid, a piece of DNA that encodes for an Endo III that can't do the fixing reaction, but it still has the iron-sulfur cluster in it, that fixes it too. It can do the DNA signaling. And then what Amy Boll found was if instead she generated a mutant that did have the iron-sulfur cluster, could do the fixing reaction, but it couldn't do the electron transfer. It had a lousy and insulating protein-DNA interface. So it couldn't do the electron transfer, and when you put that plasmid back in the cell, you couldn't have a decrease in the number of mutations. It wouldn't work. So we now have this relationship between Endo III and Mut-Y and their efficiency in reaction based on DNA electron transfer. So we think we're building a case that this might actually be a way to achieve signaling, long-range signaling between proteins within the cell. And so now when you start thinking about it, there are all sorts of possibilities to get information disseminated across the genome, across those billions of base pairs of DNA, using the DNA sort of like a wire. So that's where we are right now. These are the examples that I told you about of proteins doing this DNA-mediated signaling. Every day, when my group reads journals, every day there are new DNA binding proteins that we're finding that have iron-sulfur clusters in them, that have redox active groups, where we could imagine the possibility. We don't yet know. We're now looking at as many cases as we can that nature might use this long-range signaling to regulate expression, to respond to stresses within our genome. So what I want to leave you with is the following point, which I consider to be the most important point of everything I've said. And that is what I've been telling you about today has been essentially a 20-year basic research program. And we had no idea when we started these experiments that I'd be telling you today about genomic signaling. This started out as a very simple esoteric experiment in the lab, taking little metal complexes, adding them to a solution of DNA, and seeing if we got fluorescence quenching, electron transfer. From that, we said, well, gee, maybe this works. Maybe that's important for how DNA gets damaged. And we showed that the metal complexes could damage DNA from a distance. And then we said, well, gee, maybe nature might use this to signal damage. And we showed that you could have holes, essentially, that you could turn on proteins in response to damage. And now what we're saying is maybe even these two proteins can talk to each other through the DNA using this kind of chemistry. And I think that's really what characterizes a lot of basic research. You don't know the answers to the questions before you ask them, and things that may start out seemingly quite esoteric end up in a different place. But if you understand the chemistry and go from one step to the next, that leads you to some interesting consequences, and necessarily to the next interesting question. This is my research group. This picture is a year old, but I thought I would show you this picture because it had a lot of the players, a lot of the people, my coworkers that have worked on this chemistry in it. And at Caltech, I have the great privilege of working with an absolutely outstanding group of students who get me excited every single day and ask really interesting and tough questions, and they're smiling most of the time. So Elon Gorodetsky and Jason Slinker have been working on this, and Natalie Murin have been working on our new DNA chips. There's Joey Generu. He just finished his PhD, and he's now at Scripps. He's the one that walked in and said, Jackie, are we saying something crazy? Can I calculate whether or not this is a reasonable idea? There's Paul Lee, who did that heroic experiment trying to activate SOX-R in a glove box. And Pam Zontz, who carefully counsel the proteins bound to DNA by AFM. There's Amy Bowl. She got finished her PhD, and she's now at Northwestern. She had the courage to bring genetics into my lab and to carry out the experiments, I think, that are leading us to really show that this may actually happen within the cell. So I want to thank all of these people for their extraordinary work and for making this all really interesting. Eric Ullman's going to teach us about spectroscopy and see how fast these electron transfer reactions go. Christine Romano's taking over from Amy in terms of these genetic experiments. And Tim Moy is looking at new DNA binding proteins, and he's already got some great results. And then other people here have done other kinds of experiments. So I want to thank them very much for their efforts. Thank all of you for your kind attention, and I would be very happy if when we're finished and the lights go up, if you come up, I'm happy to answer any questions that you might have. Thank you very much.
This program is brought to you by Caltech. Visit us at caltech.edu.